Hey, what's up?
Hello, can you hear me?
Oh, I can't hear you.
It might be me. Give me a second.
Oh, this is my fault, I think. Hang on. Okay, say something now.
Is it my fault? It might be my
No,
fault.
no, it was my fault. I fixed it.
Oh, cool. Okay.
Yeah, yeah. It wasn't my fault. It was my computer's fault. Never my fault.
True.
I'm kidding. Okay, yeah. So the first thing that I was doing here while I was waiting on you to get in is just looking at that again, that mechanism that we were talking about the other day. I got a better picture of it. This one is out of the paper that I wrote. So I guess I'm like very biased because I made it, but and it's kind of pixelated a little bit because I made it like 10 years ago. But anyway, so this is just like I think it's a simpler picture of what's going on. There are parts of it that I need to like cut out of here because they're like not MCAT relevant, like this U46619 thing. That's like the molecule that we were studying. I don't think that the type of receptor is necessarily relevant either, but everything else in this is because this is kind of like how vascular contraction takes place. But you can imagine like anything being downstream of all of this that's going on. But so like the part that you were interested in the other day is like everything prior to this line. So
Sweet.
just talking about
Okay.
like how calcium comes out of the sarcoplasmic reticulum, or in the mechanism that you had, it was saying endoplasmic reticulum. But either way, so I
They
was
do mention like later on, or at least on the right side, so where you have like the RHOA GTP and
yeah,
downwards. Um, they do mention like other not non-specific, but like other phosphorylation
right,
cascades that
yeah,
like
yeah.
may be
So other
affected.
things, right, right. So other things can happen, but basically, it's just like that whenever you activated, pretty much anytime you activate a G protein, and I did some reading about this because I definitely needed to review it. I hadn't really thought about any of these pathways in a while. But it's really anytime you activate a G protein, it's going to split the alpha subunit from the beta gamma subunit. So that's the first thing that happens. And so what I was going to do, and I can do this later so that you don't have to like watch me do it, but I was just going to write out like the individual steps over here. So basically saying like first thing that happens is G protein gets activated, which leads to the alpha subunit splits from the beta, whatever. I don't know. It's not going to let me do beta, gamma, beta. Yes, I'll just do this beta, gamma subunit. And then the beta gamma subunit tends to stay like along the inside of the membrane, whereas the alpha subunit will like take off and go do other stuff. And so looks like what's happening here is, and again, I'll write this out for you because I remembered to do it like right whenever I was getting in here today, which sucks, but my bad on that. So it's basically like the beta gamma subunit is what will like, oh wait, never mind, I said that wrong. Oh no, I said it right. Okay, yeah, the alpha subunit goes into the cytosol and the beta gamma subunit stays on the membrane. And so the alpha subunit is what goes and activates that phospholipase C, and that causes the cleavage of PIP2 into IP3 and DAG,
Okay.
like IP3 and diacylglycerol, and then those go and do their separate things. And I'll write all of this out for you because I've literally got it written right here. I've got it typed over here. I just don't want to I don't want to paste the whole thing. I mean, I can, but it looks like this. So like the whole thing is probably just more than you need because it's like that. So I'll condense it down into like individual steps and make it a little more user friendly.
Okay.
So anyway, so I just wanted to show you that I was doing that. And if this one looks better, I can do it on this, or I can do it on the one that we were looking at the other day, which I think, yeah, it's right here. So,
Sweet.
I mean, I
Yeah.
guess it's the same thing.
Yeah.
Yeah.
Same
I think what
thing.
was like, yeah, I think
It's
what
just
maybe
different.
and it may not have been throwing you off, but I think what was throwing me off was I didn't recognize that this and this were the same thing the other day. I was like, what is going on here? Whenever I was looking at it. So it's basically like this thing just moves over here, and that's what they're showing.
Yeah.
Yeah.
They kind of just completely ignored the beta and gamma subunit. It just disappeared.
Yeah. Yeah. It just goes and does other stuff, which is not as relevant. Like the alpha subunit matters more. But yeah, just FYI. So that's the correction from the other day that I was kind of like having a hard time reconciling. Is like I wasn't recognizing that was the same thing. So anyway, I'll do it just like as soon as we get out here today and send it to you. That way you don't have to like watch me do it right now, because that's kind of boring and not a good use of your time. So anyway, so how many questions were you able to get through? I was simultaneously pulling up all of my faculty
Oh
portal
my
stuff
God.
and all of that.
I don't think I
Um,
summed them up, but I did do them every day.
oh,
I will
nice. Good job.
say there were some days when I did like 25. So I didn't
Okay.
consistently do 30, but I did do them every day, and that's
Nice.
better.
Cool. Oh
Um,
yeah, I'm looking at it now. Yeah. Okay. I recognize what I'm seeing now. Yeah. It
yeah.
says, yeah. So, okay, cool. Good job. Nice work. No more
Oh,
shame.
yeah. Well,
Yeah.
shame in terms of I'm not doing well on that front, and I do recognize why, given I haven't memorized like half of the stuff I need to in order to like answer some of these questions I've memorized, some of which I'll ask
Okay.
you about today. But that is frustrating.
Yeah, and it looks like you're focusing and we talked about this, you're focusing on the biochem questions. That's
Yeah.
the section that is the steepest in terms of memorization, at least in my opinion.
Yeah.
So,
I'm
makes
still
sense.
kind of navigating what is the best way to memorize it for myself. So I'm like working
Yeah.
on those study guides, or like little like 1 page summary sheet of basically like every chapter, or not every chapter necessarily, because like for metabolism, each metabolic
Yeah.
reaction has its own sheet, but that kind of thing. And I think the next I just have to be flashcards, even though I hate them with a burning passion, but
Yeah.
I must.
Yeah. No, it makes sense. Yeah, you can do flashcards, or something that I've told people to do in the past, and I don't know if this will help you. I may have mentioned it, is and this is more for people who are bad about making like spending forever making just beautiful study guides that they're like look like they're trying to publish them. Is that I've told people to like take like printer paper. I liked paper because notebook paper would like the lines, it just annoyed me. So I would just go steal paper out of the printer at my campus library. Like I just like wait till nobody's looking, and just like open the thing up and grab a bunch of paper and stuff it in my backpack and leave. So that's not the advice, but but if you take like printer paper and you just draw this stuff out and write it down over and over again and then just throw it all away at the end,
Hmm.
that makes you be kind of efficient, because you know you have to throw it away at the end.
Hmm.
And it's kind of just like a good, like iterating it over and over again, writing it down over and over again can be helpful for memorizing stuff. But it's up to you. That's just something that I found helpful for myself in the past.
No, I have
For
now moved on to printer paper, which is lovely, because I
nice.
feel less guilty about using really nice notebooks.
Oh,
I
okay.
don't know why that is a thing, but it is a thing.
That's funny.
I do, however, need to get better about repeating that process, because I feel like even though I'm condensing it essentially into like 1 page, I still either one want to make it look nice, or two, am adding like way more information than I like
Then
feel
you
like I
need to add.
should be. Yeah. And in the
Yeah,
end, I'm like, this is just a paper written out completely about
exactly.
this one subject. And like, that's
Uh,
not what I need to be doing.
Yeah, yeah, because in reality, like, are you really gonna go back and look at it that much? I mean, if you are, that's one thing, but I know for myself, it's like the act of writing it down is more important than having written it down,
Hmm.
than having the
Hmm.
paper. And so this sounds like it might be the perfect tip for you then, because if you're spending too much time making it look neat and stuff, then just telling yourself, I have to throw this away at the end. Like you
Yeah.
have to throw it away at the end or recycle it. then like that is the automatic solution to that problem, because if you the first couple times you throw away something that you spent time making look really nice, make you stop doing that. And so like that was for me, especially like studying organic chemistry, that really helped me study OChem whenever I was in
Yeah.
undergrad, starting to do that. And then I did it a lot in anatomy in med school, just like draw stuff, throw it away, draw stuff, throw it away, because it's more useful. The like act of making it is more useful than like having it and reading back over it,
Yeah.
personally anyway.
Yeah.
So you might try that. It's not fun, but you could try that.
No, but I think that's like my goal for these next few days. Like starting on Thursday, I'm just gonna make myself do that, honestly.
Yeah.
Um,
Throw it in the trash and dump water on it.
oh my god, I don't need to do that. It's already
I'm just
constantly
kidding.
raining here.
Oh, okay, there you go. Yeah, throw it in the trash and then
Nature
put it
will do that
outside.
for me. Yeah.
Okay, cool. Yeah, I wish it would do that here. It's always like 80 and sunny here. I hate it. It's so annoying.
I won't even open that conversation again.
Yeah, okay. It sounds good. Oh, I want to. I might get your opinion later. So I'm looking at buying some snow skis. So when we finish here, I might get your opinion on like snow
Oh
skis
my
because it
god,
sounds like
yeah.
you might be better at skiing than I am, which is something that I don't know a lot of people who are. So,
Oh my god, yeah. Show me new skis. It's
okay.
been so many years since I've
Oh,
had to buy new skis.
oh, really? I've never owned any skis. I've always just rented them whenever I go. But yeah,
Well, at least around here, it's like way more expensive to do that than like
right.
have a set and then like, I mean, it's a really shitty set to be quite frank,
Uh-huh.
but like consistently revamp them somehow and then have
Yeah.
them for a bunch of years.
Yeah, if I was going as much as I would like to, then it would definitely be more efficient for me
Oh,
to.
fair enough. Yeah.
But yeah, I like when I went last spring break and I haven't gone since then. And prior to that, I hadn't gone in like 5 years, which is just pathetic because it's like my favorite thing to do. I just need to make myself go do it. But I always have a hard time like justifying spending the money and like taking the time and all of
Well,
that.
there's no
So
skiing in Texas, right?
no, I have to go far. Like I
Yeah,
always
you
do.
have to like travel.
Oh yeah, it's an expensive process because you have to find a place to stay and like where we like to
Oh,
go to Breckenridge in Colorado, and the ski lift tickets there are
oh,
just
my dream.
extraordinary costs. Yeah. Yeah, it's like $180 a day or something like that for a lift ticket. So,
Oh, good God. No, no,
yeah.
no. None of that on my end.
Oh yeah, yeah. Yeah. No, I that's why we don't do it very frequently.
Yeah,
But
that makes so much
when I was
sense.
in med school, when I was in med school, I felt like I was rich because I would take loans and just like live off my student loans, which is not a good choice. But
Never do.
yeah, but I always felt like I had a bunch of money then, so I would always buy a season pass when I was in med school, but I hadn't done it since then. So yeah, this is
I
like,
need to do that one day. That is one thing I also have never done because I don't go enough, even though I live close by. But
oh yeah, yeah,
one day,
yeah,
one
yeah,
day.
exactly. That's why you're being a doctor, so you can make money, right about that.
Yeah, literally my only reason.
There are better ways to make money, I promise.
That's what I was. Oh my God. I had that conversation recently. I was
Yeah.
like, you would think that if I was choosing to do this or choosing to make money, I would have done like
Mhm.
20 other things or like not even
Yeah.
gone to college and just done computer science. Like
Yeah.
there's no need.
Right. Yeah. If
Effort.
money is your goal, you're in the wrong place. So yeah.
True.
I mean, you will make a bunch of money as a doctor, but it takes so long, like the effort. It's like
Yeah.
you can
Also
get
kind
more
of
money.
depends what you end up doing or like wanting to do. Because there is
Oh
like a
yeah.
massive disparity.
Oh yeah, for
Um,
sure. Like my
I don't know.
girlfriend's a pediatrician and they make like 250, 240, something like that.
Yeah.
And then I have other friends who are like in neurosurgery and they make seven figures. So yeah.
Yeah. It's
Yeah.
crazy.
Yeah. Yeah. But they also work 80 hours a week. So it's whatever you want to do with your life, I guess.
Yeah.
Anyway, so do you want
I
to do
could rant
MCAT stuff?
about that forever instead of doing
Yeah.
MCAT stuff. But yes, I
Yeah.
do want to do MCAT stuff.
Okay, cool.
Um, uh, I have pretty much a question from like each of the tests that I've taken,
Okay, cool.
or like little practice problems.
Yeah, just
So
I've got them all pulled up here. So just tell me
oh
which one you want to start on and I
sweet.
can click on them.
Can I still tell you the Whoa! Did you see me disappear completely?
Yeah, you have disappeared.
I went purple. I don't know what
Oh,
happened
there
there.
you go.
Oh well.
You're back now.
Um, okay. Let's start on question 401979, and I can give you the test ID if I can.
Yeah, which day did you take it? Do you know?
Oh, yes. Well,
Or
can I
what
give
like
you the
which
test ID actually? Because I wrote it down, and that would be
Yeah.
easier.
Oh yeah, yeah,
Okay,
absolutely.
sweet.
That works
I
too.
was trying to find the test number, but I can't. Okay, the test ID is 246701793.
246701793.
Yep. And the question ID is 401979.
Which question number on the test? Do you know?
Oh, let me find out.
401. Oh, wait, I can see it. Never mind. 401,979.
Yes.
Okay, found it.
Wait.
All right. All right. So it says under physiological conditions, peptide bond formation requires coupling to GTP hydrolysis. I always forgot that I always forget that it's GTP and not ATP in peptide bond formation. It's funny. 'Cause I'm
I
making a
also
video about
forgot
that.
about that, but
Yeah. No. All right. So, yeah. So GTP hydrolysis, peptide bond degradation does not require GTP hydrolysis, but does require protease enzyme to proceed at physiologically relevant rates. Okay, I know where we're going here. Based on this information, under physiological conditions and in the absence of proteases, the peptide bonds that form during protein synthesis are okay. So why would something require GTP hydrolysis or not? Like what's that telling you? One thing does require it, and one thing doesn't require it.
um, you require hydrolysis in order to break a bond.
Okay, so
Um,
what it's saying though is like let's just say that we have two amino acids. So let's say like I don't know, two that I just think of right now are like glycine and alanine. And so
mhm,
to bind those together, we form this peptide bond. And so forming it hang on. So whenever we form that bond, we can't just go glycine plus alanine equals our like peptide bond, right? We have
mhm.
to do glycine plus alanine plus GTP. It gives us our peptide bond plus GDP, plus PI. Why do we have to do it like that? Why can't we just do the part without the GTP?
Because it requires energy in order to form or break the bond.
Yeah, exactly, because you need energy input versus we can go glycine, alanine, peptide and take that and then form glycine plus alanine. That does work. So why don't we need to couple this one to GTP?
Um, why don't we need to couple this one to GTP?
Yeah.
Because they're separate. I mean, you're breaking the bond, so it is lower energy.
Okay.
And you don't
Yeah.
need like an input of energy in order to break the bond.
Right, exactly. So basically what it's saying is like in this direction, in this bottom one right here, when we're
Uh-huh.
going that direction, our delta G of this reaction is going to be less than 0.
Right. Okay.
Whereas if it's less than 0 in that direction, then that means if we reverse it, then our delta G has to be greater than 0 in the reverse direction because you just flip the sign, right?
Yes.
And so if our delta G is greater than 0, that is never going to happen unless we couple it to something more favorable.
Mm-hmm.
Okay. So what that's telling us then is that our reaction is thermodynamically unfavorable in the bond formation direction and thermodynamically favorable in the bond breaking direction.
Yes.
Okay. All right. So that's the thermodynamics dynamic side of this. And so then we think about kinetics and so what in the question stem gives us like any indication toward kinetics?
The fact that it requires a protease enzyme in
Okay.
order to proceed.
Yeah. So that tells us that without the enzyme, the reaction's still going to happen. It's just going to be slow as shit.
You're right.
Right. Okay. Okay. So yeah, so that's just like my consideration before even looking at answer choices about this whole
Yeah.
system. Yeah. Okay.
And
So
I
let's
agree.
see.
I had a similar thought process
Okay.
when looking at the question.
All right, cool. So then it's asking us based on this information under physiological conditions and in the absence of proteases, the peptide bonds that form during protein synthesis are. All right. So we think both thermodynamically and kinetically stable. I don't know what it means when it says kinetically stable, to be completely honest. Like that just sounds weird to me. Yeah, but yeah, so
I
under
think the
physiological.
like an intermediate, if it has like an intermediate, for example, it is just simply less energy to be in. Or like I don't know. I actually don't
Okay.
know.
Yeah.
No, I'm questioning
Okay.
it.
Okay, yeah. So but from the thermodynamic side of it, we can say that the peptide bonds that form during protein synthesis, they are thermodynamically stable, right? Because to
Mhm.
break them would require energy input, right? Or wait, no, I'm sorry. No, to break them does not require energy input.
To break them does not work. Yes, I agree with
Okay.
that.
So they're thermodynamically unstable then, right?
Yes.
Okay.
Okay, bonds
So
that form. Yeah, would be thermodynamically unstable.
so they're thermodynamically unstable. So that means we have to pick that one or this one, right?
Mm-hmm.
And then we have to think, okay, so then we consider the kinetic side of this. And so I think what it means by kinetically stable is that thing, and this is that even though this thing is thermodynamically unstable, it's able to exist. We're able to isolate it, right? It doesn't just break down automatically because the activation energy is so high that we need a protease, we need an enzyme to make the bond actually break, even though it's favorable for it to break. It just
Okay.
happens so slowly that it's stabilized. So I would say here that B is the right answer because even though it's thermodynamically unstable, it is the bond itself is stabilized by its kinetics. The fact that it's
Mm-hmm.
unfavorable, so it's favorable, energetically favorable to break it, yet it doesn't break because that reaction proceeds so slowly in the absence of those proteases.
Because the reaction is okay. Okay, yeah, I understand what you're saying. I My thought process behind that one was kind of flipped from, but like I agree with with what you're saying now, and I understand it from that point of view. But I think mine was because it proceeds so much better with the enzyme, it is more unstable as a result if it has to proceed without, in like the
Okay,
absence of proteases. Um, but you're saying that's not
gotcha.
the case.
Yeah, yeah, yeah. I'm saying that that the bond itself is stabilized not because it's energetically stable, right? Because it's the breaking it is favorable from a delta G
Mhm.
perspective, right?
Right.
But it's yet it is stable because it just takes so much activation energy to break it that without a protease present, we just can't.
Right. It's just slow as shit, but can
Yeah, yeah,
still
like
proceed.
yes, yeah. Yeah, it'll
Okay.
still proceed. It just does so slowly that we don't observe it happening because it would happen so slowly.
Okay. That makes sense.
Okay, cool.
But with
And I
like
just
kinetically unstable things in general, then would they just not be able to proceed at all without the presence of like a specific enzyme or whatever, like coenzyme, for example?
So what I would say let's say that we ask the same question, but proteases are present now,
Mhm.
then I would say that it's still thermodynamically unstable, right? Because delta G isn't changed if we add the protease. And then once we add the protease, I would say it's also kinetically unstable because now that the protease is there, the enzyme is that bond is no longer stabilized by the slowness of that reaction because the protease speeds it up sufficiently that now the bond itself is just unstable and it breaks.
Oh, interesting. Okay.
Does that make sense?
No, that makes perfect sense, but I also like want to disagree because it is with a protease stable in my mind is that like it proceeds as it's supposed to,
Yeah, so you're
which
talking
like is
about it from
probably not the same thing.
you're taking the perspective of the reaction, but it wants you to
Mhm.
take the perspective of the bond itself.
Oh,
See,
okay.
it's because it says peptide bonds that form.
Mhm.
Yeah.
Mhm.
Yeah, because a reaction can't be stable or unstable. A reaction is a process.
Right.
The bond itself or the molecule or whatever is the entity that is either stable or unstable. And so what it's saying is
Okay.
this whole thing, like this, the glycine alanine bond, for instance, it's saying, is that going to sustain itself into the future? If it is, then it's stable. If it's not, then it's not.
Okay. That makes a lot more sense now.
Okay, cool. Good.
Yeah.
That's the goal.
Yeah. My thought process, I guess, was just flipped on that front.
And it
Again,
looks like,
perspective. Last week
yeah,
was
yeah.
perspective.
Yeah, I
Okay.
think I'm starting to realize over the last that's kind of like my theme of the last week as well, actually, as I work with people, because I'm starting to realize that makes people get stuff wrong more than I would have guessed before. And it's not something I've ever heard anybody say before. So maybe I've got like a inside track
Maybe.
on this.
I don't
Yeah.
know. I always thought it was my focus that is the issue, but regardless of how hard I focus, I still flip the wrong perspective. So I don't know what it is, but
Yeah, I think it's probably just it's just like paying you could call it focus, but I think that is just a more general way of saying it. You know, like it's not quite putting your finger on the problem.
Yeah.
Yeah.
Cool.
And it looks like I'm right. I just clicked the answer and submitted it. It says that B is correct. It also says that only 34% of people got this right, which tells you that it's a really hard question.
Fair enough. I think with the funny is the lower scores are always the energy related ones, which is kind of funny.
Yeah, because they're the ones that are going to require you to integrate a bunch of concepts, and I think people have a hard time with that.
No,
That's
I
my guess.
think those are the most fun ones.
Nice, yeah. Well, it's because you're also like really quantitatively oriented. It seems, like you're
that's
good at
fair.
math. Yeah,
That's fair.
yeah.
Well, less good
Which
at
is why
reading.
I say that I truly believe that you can do as well as you want to do on this test. You're really smart. So yeah.
I'm trying to buckle down. I am buckling down.
Okay.
Yeah.
Nice. Yeah. I like that.
I am buckling down. Okay.
Yeah.
Next
buckling
question.
Down.
Speaking of enzymes, there are so many freaking different types. And for some reason, in the last week that I've been doing all these practice problems, I've encountered a multitude of questions that have asked me specifically about like what type of enzyme this XYZ is in like a specific problem. So I was wondering if you had any like random review material, or not really, or if we could review the types of enzymes.
Yeah, for sure.
Because there's so many.
Yeah. It's funny you mentioned that because I'm actually trying really hard lately. And I've been speaking of buckling down. This is the thing that I need to force myself to do, making this like video series on enzymes, basically. And my social media person tells me that I have to make them all under 2 minutes so we can post them on Instagram, which feels impossible. So that's why it's hard. Yeah, it's kind of ridiculous, but making these whiteboard videos. Anyway, so I've been thinking a lot about this. So yeah, for sure. We can definitely talk about it.
Yeah, I encountered like zymogens for the first time. Zymogens.
Zymogens is how I would say it.
Zymogens.
I'm cool with however you want to say it.
I've been saying it zymogens.
That's fine too.
I don't know.
I'm Googling that really quickly because I've honestly forgotten what a zymogen is. Inactive substance which is converted into an enzyme when activated by another enzyme. That doesn't make any sense. Oh, okay. I know what a zymogen. All right, cool. So like trypsinogen turns into trypsin. So it's like a proenzyme. I don't know why they don't just call it a proenzyme. Okay, it is a proenzyme. It's a synonym.
It's a type.
That's Wikipedia.
Oh, nice.
Yeah. So have you heard of a proenzyme?
Proenzymes are the ones that are like the precursors to like the end molecule, so to speak, the ones that actually do things.
Yeah, for sure. Yeah, I think that here, I'm gonna go and write this. So zymogen, however you want to say it. I'm pretty sure it's zymogen, is the same thing as a proenzyme.
Okay.
And the pancreas is the best example to think about for thinking about this. So, and I'm just gonna write, let's just say like inactive form of an enzyme which can be activated by some chemical process. It's normally like cleaving of it. Say like normally cleavage of some, say like inactivating region of the molecule. All right. And so, yeah.
Sorry, just to clarify, you say it's the inactive form of the enzyme, meaning.
Yeah. So here's the best example.
Yeah.
Okay. So let's say, which one should we use? Let's use hang on. Sorry. I just want to make sure I give that my example actually works. Dang it. I can't type. Okay, cool. I was right. All right. So trypsinogen. Trypsinogen is that's spelled wrong. Trypsinogen is the proenzyme of trypsin. And that just means it's like the inactive precursor form of trypsin. And so the reason that you do this is because trypsin
Hmm.
is an enzyme that degrades proteins. It like breaks peptide bonds. And so if you start releasing this enzyme accidentally or too early or in the wrong place, you're going to melt yourself and you're going to die. Okay, so
Okay.
the disease pancreatitis is a really good example of this. So the pancreas produces most of your digestive enzymes, and these digestive enzymes are really good at breaking down proteins, like I was just saying. So they'll cleave peptide bonds. The problem is that most of what makes you up as a person are peptide bonds, are proteins. They carry out most of the functions in the body. And so if you release trypsin, if you're just making trypsin, the active form, then it's just going to start tearing shit up. And so instead of making the active form, you make it in its proenzyme form, which is storable, whereas the enzyme itself, trypsin, you can't really store it. And so you make it in the form of trypsinogen, which is like, so we'll just say that it's like this peptide. Let's just say that's the peptide in its like elongated form before we like wad it up. And then you release it as trypsinogen. And then in the duodenum or duodenum, however you want to say it, it gets cleaved into trypsin, into the active form by just like cutting part of it off. And as soon as you cut part of it off, it's able to fold into its like native form where it actually is able to cleave these peptide bonds and digest the food in your GI tract. But if it gets cleaved into trypsin while it's still in the pancreas, then it will start to digest the pancreas itself and you die. And so that's like
Nice.
what pancreatitis is. Yeah.
Okay.
Yeah. So you'll generally like release a lot of enzymes. Most digestive enzymes are released in a proenzyme form so that they can be activated only in the right like physiological compartment, if that makes sense.
Okay.
Yeah.
That, yes, that does make sense. And all of these are not rather, it's never all enzymes, but they are usually involved in cleaving or just activating this next molecule that
Um,
proceeds.
could you say it a different way? Sorry. I just want to make
Yes, I
sure.
can say it. So for example, I think the other two that were involved were apo enzymes and holo enzymes, which
Yeah,
I
that's like
think
a different
are,
thing.
oh, so also not proenzymes.
Right, right.
Okay, then never mind. Um, under what would then my okay, wow, words, 1 second. If proenzymes are like an overarching factor, then what would be a different type of proenzyme?
Um,
If
proenzyme is so proenzyme is just like a big long peptide chain that when the inactive part of it is cleaved off turns into the active enzyme. So proenzyme is like an inactive precursor of an enzyme.
Oh, okay, so I just misunderstood. Never mind. I was
Yeah, no
under the
worries.
assumption that proenzyme was like the overarching type of enzyme that
No,
zymogens
yeah,
were.
yeah.
So,
Proenzymes generally don't do anything.
okay.
Yeah, but zymogen is the same thing as a
Mhm.
proenzyme.
Right.
Zymogen, proenzyme, these things are like enzyme precursors. You cut them into enzymes.
Okay.
Yeah.
Understood.
Okay, cool. So it's like there's also an example of this for non-enzymatic proteins, like proinsulin is an inactive form of insulin that like is and that's what's released by the pancreas also, coincidentally. And it once it gets in the blood, it gets cleaved into insulin, which is like the active version. And so you just like cleave off some inactivating sequence that exists in the molecule, probably just so that it doesn't start doing its job too early.
Okay. So
Yeah.
then,
So So it's an inactive precursor form of the enzyme, is a proenzyme or a zymogen.
okay, cool. And then holo and apo enzymes, just to track back to that and clarify. Um, they're not, they don't function as um, proenzymes, or they're not a type of proenzyme or something, but exist as separate enzymes that are basically the same, just with or without cofactors.
Yeah, exactly. Yeah, holoenzyme is like the protein form, and a coenzyme is, or I'm sorry, an apoenzyme is the protein form, and a holoenzyme is the like combination of the apoenzyme and the coenzyme.
Mm-hmm.
Yeah.
Okay. Enzymes are all proteins, though.
No, they're not. So, okay, this is here. Let me do this.
Interesting.
This is gonna be a much more efficient way of doing this. Unless you can't hear the volume, and then it's not, and then I'll just say it. But this is literally the thing I was working on earlier today. This is, and it's just gonna be more efficient because I've been working on this for fucking ever. One second. Let me send this over to you. This is just like it is a video more or less of talking about like why enzymes are not necessarily proteins. So it's,
Interesting. Okay.
yeah.
I feel like
You
I should have known that, but I've always been
no,
working under the assumption that they are proteins.
yeah, most people, I'll bet you anything, like the vast majority of people in your position prepping for this would make the exact same mistake. Hence why I'm making this video.
Interesting.
So,
Okay.
yeah, here you go. I'm just gonna send you the link. That way I don't have to worry about you being able to see the or hear the audio or whatever.
Mhm.
It's literally like 1 minute long. I just sent it to you in the chat here.
Yeah.
All right, I'm gonna go grab a drink really quick and use the restroom.
Okay.
I'll be right back. This is like the second minute of it has no animation to it yet. This is just like it as it is right now. So bear with me,
Okay.
but
Can you
I'll be
hear
right
it
back.
playing? Or no?
Uh,
Wait.
no, I can't hear it. I can barely hear it, but I'll be right back.
Okay, cool. Wow. I literally just finished it.
Okay, cool. So did that make sense?
Yes.
Okay, cool. So
Not even ribosomes are the proteins.
yeah, no, ribosomes aren't proteins. Ribosomes
Oh
are
my
made of,
god. I'm enraged.
yeah, so ribosomes are made of rRNA, which comes from the nucleolus.
Oh no. I
So do
need
you,
to review that further.
yeah, so if you go to like the maybe let's see, like the 30-second mark, and I can also just pull it up
Mhm.
since I'm screen sharing,
Mhm.
like this part of the thing where
Right.
the thing looks like an egg yolk. That is the nucleolus, the thing that's in the very center of the nucleus.
Right.
Yeah, and that's where our RNA comes from.
Right. Okay.
Anyway, so does that kind of make sense? Like, so enzymes are like 3D biological macromolecules that just provide the right conditions for a reaction to happen more easily. And that's where
Yes.
I'm ultimately going with this. I'm just trying to figure out how to like add an animation to the to the rest of it, obviously, because that took me so damn long to draw all of that.
I
Yeah.
can't believe
It's
you drew literally all of that.
oh yeah, I drew every last bit of it except the picture of Kevin Durant.
Yeah, I was just gonna say, I can't believe you chose Kevin Durant as the MVP.
Well, so I do not think Kevin Durant's necessarily MVP, but that quote where I said like enzymes or ribosomes are the real MVPs or something,
Yes.
that is a Kevin Durant quote from when he won the MVP in like 2015 or something like that. That's a picture of him when he won the MVP that year.
Wait, actually?
Oh yeah,
That's
yeah.
incredible.
Because in his speech he was crying and he said talking about how his mom raised him in a single parent household, and he said his mom was the real MVP. So he said, "You the real MVP," and it's a quote that like
Oh,
maybe 20% of people will get tops, but it'll definitely earn me credit with that 20%.
I think
Probably
so.
all guys, but yeah.
I didn't catch it, but I really thought he said ribosomes were the real MVP for a second. I was like,
Oh,
wow,
no, no,
that's
no, I'm
really
sorry.
wildly
He's
impressive.
saying it about his mom. No, not the ribosomes for the real MVP. I bet you he doesn't know what ribosomes are. I would be shocked.
Maybe once learned them, but not anymore.
Yeah. So according to my brother, the only thing we gotta have to worry about that video is that picture right there. But I'm like, ESPN doesn't care. So
The amount of like memed videos of Kevin Durant and every other basketball
right.
player ever on YouTube They
Yeah,
won't pay
exactly.
attention to yours.
Yeah, that's what I was saying. Is like if we get to the point where we have to worry about that, that's a good problem to have, in my personal opinion. So, yeah,
Mhm.
not too worried about it right now.
Well,
So, but yeah,
I
I drew
enjoyed the
everything
video.
else because we're worried about stealing images from from like actual stuff. So yeah,
I I don't even understand that, honestly. That
what like?
beyond my ability. Well, because like, everyone uses this vaguely same
Mm-hmm.
pictures of a cell or draws it
Oh
pretty
yeah,
much the same exact way.
I
So
definitely
why
looked
would
at
you get sued?
those. Yeah, I definitely looked at other people's images when I was drawing mine, but no, I just figured might as well not have to, might as well just do it right and then not have to worry
True.
about it later. So yeah,
Very true.
that's why I did it like that. Anyway, so yeah, so all of that to just say that also there was that bit about Rosalind Franklin stealing the DNA structure, but I thought that was funny too. But so all of that just to say that enzymes are not necessarily proteins, though most enzymes are proteins, that
Mhm.
ribosomes are actually like the OG enzymes. And those are, and I'm sorry, not ribosomes. RNA is actually the OG enzymes. Yeah,
Cool.
because RNA was like the OG life. So it had to
Oh,
do everything.
total tangent, but now that we talked about ribosomes,
Uh-huh.
can we possibly go over ribosomal structure?
Yeah, sure. Yeah. So we
Um,
could use that
because
also.
there's a lot of subunits.
Yeah, yeah. So there's really like depends on if you're talking about eukaryotes or prokaryotes, but there's like so the one that I drew there was eukaryotic, and
Mhm,
so you've got like the 40S and the 60S ribosomal subunits. And so the 40S subunit is what comes in and recognizes the sequence on mRNA. And so it'll find that like original methionine signaling part of the mRNA,
mhm.
the AUG, the start signal. And
On
so the
the
4
mRNA, you said?
yeah, yeah,
Okay.
yeah. Yeah, so like on not to just like keep going back to the video, but it is an easy way of looking at it just because I've already drawn it. But so yeah, so the way that this happens is the like all right, hang on. So here. The all right, so see I wrote 40S ribosome right here on this little like
Mhm,
weird looking thing.
mhm,
And so then the 40S ribosome will be the first thing that binds to the mRNA. And so it recognizes AUG sequence on the mRNA, which is like the start sequence, and it binds there. And then the 40S ribosome is what recruits the big thing, the 60S ribosome. That's the one with the EPA sites on it.
mhm.
It recruits that in and binds has so the 60S ribosome comes in and binds to the 40S, and those two together for some idiotic reason I can tell you why, but the 40S and the 60S once they come together, that's what forms the 80S ribosome, not the
Okay,
100S ribosome, the 80S ribosome.
okay.
Yeah, and that has
80.
to do with something with their like what they do, where they come out in a centrifuge or something like that. Because the S stands for sedimentation
You'd
factor.
think it would be 100, but that's fine.
Yeah, I know. Yeah, so whatever. But yeah, they come so
Okay.
40 plus 60 equals 80 for eukaryotes. And then for prokaryotes, those numbers, it's the same idea. It's just different numbers. I'm gonna look it up really quick because I don't remember. Um ribosome. Okay, so for prokaryotes, it's the same except that 40 is now 30 and 50 and 60 is now 50. So it's just like 10 less on each of them. And those come together to
Wow.
form the 70S and not the 80S.
Okay, but that makes sense. 70s? That's
No,
like,
because it's 50 plus 30.
oh,
Yeah.
no. Okay.
Yeah, I know. That's that's ridiculous. And so I will make a that's why I said somewhere in there like that's a different story for another day because I will make a translation video at some point, but I didn't feel like it right then, even though I
That
practically
was so
did.
much drawing.
Oh yeah, especially when I like have to animate it and like make the tRNA and then it like
Right.
scoots over. It's just yeah. So yeah, and so then once they form that peptide bond, the whole
Mhm.
thing, as you can see in this video, will shift. That shifts, and then the other one shifts, and then the
Right, and
next
it goes
tRNA comes in.
from A to E, exits out
Uh,
of E, or E to A.
yeah. So it goes. Yeah, it goes the way that it. So it starts out the way during initiation. See down here
Mhm,
where it says at the very bottom of the screen, it says translation initiation. The methionine residue, the first one that comes in, binds to the
mhm.
P site. in the middle.
Right.
And then once the thing shifts, so that methionine stays in the same spot, it's the ribosome that shifts. It will shift so that the methionine is now in the E site. And then once it shifts again, the methionine's gone.
Whoa, okay. I don't know if this is, I think I just learned this wrong originally, or maybe remembered it incorrectly. What is it, like no, it's not the butterfly effect. Anyways, it's the ribosome that shifts. It's not the sequences that are shifting over into the next site.
Correct. Yeah.
Oh my God.
Yeah. So the
Okay,
ribosome
cool.
shifts along the mRNA. So like eventually this ribosome would go all the way to the other end of the mRNA.
Right.
Yeah.
Yeah, that makes sense because that's how like all of the other enzymes function during like transcription, for example.
Right. Right. Yeah.
Interesting. I thought it was the stable one, but that's okay.
Yeah.
Good to know. Good to know.
Yeah. So anyway, yeah, that's how that all happens.
Cool. Good tangent.
All right. Yeah.
Another one for you. Tangent to KM. There's a problem that I understand why I got wrong, but I can pull it up anyways. It's in test number 246.
All right. One
Three
second. Let me.
seven. Oh, sorry. Continue.
Yeah, I have to type it in there. Sorry. 24637.
5.
What's next?
Oh, 573.
573.
Mm-hmm.
That's all.
Mm-hmm.
It doesn't. Okay. 2463753.
375573.
375. Oh, 2 5s.
There's 2 5s.
Okay. Yeah, that worked. And which number is it?
403132.
Okay. Cool. Yeah, I'm there.
So I bring up KM because it talks about KD, and I understand why I got the problem wrong, but I'm not 100% solid on the difference between KD and KM because they're both descriptors of affinity.
Yeah. Yeah. KD is going to be the dissociation constant. That's what it normally stands for. I'm just seeing how they're using it here.
I think it's also the dissociation constant.
Yeah. Let me look really quickly how this is used. Do you know, have you looked it up in the book? Do you know where KD is found? Now Kevin Durant pops up. First thing that happens whenever I type KD.
Yeah, let me find it again. It might be in not the biochem book, actually.
Oh, really? That'd be
Let me
strange.
59 to 60. Let me see.
I think that I see. I want to say that it's the opposite of KM. It's like 1 over KM is KD. That's my intuition, but I'm not sure.
1 over KM is KD.
Yeah, that's I think that it's something like that. But I just don't remember right off the top of my head. So I'm trying to look.
You might be right. I just hadn't I mean, I wouldn't put it past me to like somehow just skim over it, honestly, but I hadn't encountered KD before. Like I encountered dissociation constants a bunch, sure. So like I understood what it was supposed to be or what it was supposed to be doing. But nowhere in the chapter 2 of enzymes did it talk about the dissociation constant.
Okay.
So that was just weird on my
end.
end.
I'm also looking though, because I haven't seen it in here either, to be honest. So I'm not completely sure. And it could be, I haven't read the passage. It could, but it doesn't look like they like introduced it in the passage or anything, did they? I don't see it other than that picture.
No, yeah. It was mostly just described in the explanation. Which is why I like understood what it was like supposed to be doing.
In the explanation of I haven't looked at that yet.
the answer.
Okay. Yeah, I haven't clicked an answer yet. Let me look at given the arcane. Oh wait, that's not the right question. Dang it. What question was it? 403132.
2.
Oh, I was looking
Mhm.
at the wrong question. Okay. Given that arcane 1 non-competitively inhibits calcineurin, which statement is true regarding the dissociation constants shown in figure 1? Okay, we can also just look at this and like try to figure it
Mhm.
out. All right, so what is arcane 1, if you don't mind me asking? Is it like an
Yes,
inhibitor?
so it's a non-competitive inhibitor.
Okay.
So it was asking like what this non-competitive it inhibits calcineurin, um, and it has the picture, which I think you can see on the side. It's figure 1. It just shows in like a really weird format, honestly,
Yeah.
what the enzyme or like the inhibitor does and like how it attaches per se. So there's 2 different ways it could possibly interact with calcineurin.
Mhm.
And you understood.
Yeah, yeah, I see what you're
So
saying.
there's a KD1 and a KD2 because there's 2 different ways it can proceed.
Gotcha.
So like I understood. I mean, in their explanation, they were talking about KD being a dissociation constant. So a smaller KD indicates a greater binding affinity.
Okay. Yeah, yeah. So it's like inverse of Km.
How is that
Oh wait, did you say smaller KD indicates
smaller
greater binding
KD?
affinity?
Yeah, which is the same as KM.
Oh, so then it's like the same thing. Yeah. Okay.
Yeah. What? What?
Yeah, it seems like it's like the same thing, or at least you can think about it that way if that's what they said. Um, did you say it said that in the passage? Sorry, or
Yeah,
did you
yeah.
say you
No,
read that in the
sorry.
book?
It said it was it says this in the explanation of the question. So like if you choose an
Mm.
answer and then it
Oh,
shows
okay,
it,
okay. Yeah.
type of thing.
Yeah, I just haven't done that yet because then I have to like submit the whole thing and then go back and find the question
No,
again.
that's
So,
fine. Yeah,
but
yeah.
and I'm also trying. I think it's almost more helpful if I think through it rather than if I just like look at the answer sometimes. So what I just realized here, I know how to get the answer to this question. I don't know
Mhm.
if it's how they said, but okay. So
Mhm.
if you look at it, it says Arcan 1 non-competitively inhibits calcineurin, right?
Yes.
So if you think about what is a non-competitive inhibitor, what's a non-competitive inhibitor do?
Um, well, it doesn't bind to the active site, so it will bind to the enzyme substrate complex.
Right. What about
Or
the empty
oh,
enzyme? Yeah.
right, or just the enzyme.
And does it have a
It's
preference?
um no.
Right, exactly. So non-competitive inhibitors. Oh, wait a second. I might be thinking through this wrong. Not so non-competitive inhibitors are going to bind with an equal. Okay, I'm right. Yeah. Non-competitive inhibitors bind to the empty enzyme and the enzyme substrate complex with equal affinity, right? And
Okay.
so what KD1 and KD2, and now I also know why it's not saying KM, and I'll tell you the difference in a second. So what KD1 and KD2 are telling you is
Mhm.
these are the dissociation constants, or you can think of them as like equilibrium constants or anything else. Basically, these constants are giving you some indication of the affinity of calcineurin for the inhibitor, not for the substrate. Because the thing
Okay.
that's changing in these two reactions is see this, we'll just call this reaction 1 and this reaction 2 because they're going this direction, right?
Right.
Okay. So in reaction 1 we have the inhibitor binding to the empty enzyme. Okay, and so the KD1 is something like the affinity of the empty enzyme for the inhibitor, whereas
Uh-huh.
KD2 is like the enzyme substrate complex affinity for the inhibitor. Okay.
Uh-huh.
And so since it told us that it's a non-competitive inhibitor, that means that the empty enzyme and the enzyme substrate complex have equal affinity for the inhibitor.
Okay.
Equal affinity. I think I said infinity, but equal affinity for the inhibitor.
Understood anyways.
Yeah. Okay. And so if they have equal affinity for the inhibitor, then regardless of whether KD is inversely or directly proportionate to Km or whatever, or affinity or whatever,
Mhm.
that means that KD1 and KD2 are the same.
Right.
Yeah,
Okay.
which tells you C is correct.
Which would happen anyways, even if you just went from the perspective of the fact that it's a non-competitive inhibitor, and by definition, it has equal affinity for both.
Yeah, yeah.
Okay.
So you don't
So
even really need a definition for KD here to get this question right, in
right.
my opinion. Yeah,
No,
yeah.
I agree. I was thinking of uncompetitive inhibitors,
Oh, gotcha. Yeah,
which, yeah, would have been one over the other. That's fine.
right.
Okay, so it's still, but like I guess I don't really need to care about KD anymore, so that's fine. But it's basically the same concept as KM in that regard.
Right.
Given that, yeah, affinity would be bigger with a lower dissociation constant.
Yeah, yeah. So and I think the reason that it's not Km is because here we're talking about dissociation of the enzyme from the inhibitor, whereas Km is a reference to the enzyme's affinity for the substrate.
Yeah, okay,
Yeah,
missed that detail. Right.
yeah.
Okay.
So Km is a comment on like how much inhibitor, I'm sorry, Km is a comment on like how much substrate do we need to make the enzyme run at 1/2 Vmax.
Mhm.
Whereas KD is more of just like directly an affinity of one protein binding something else. It doesn't matter what it is. Like, because you talk about KD with respect to like ligands binding receptors also.
Right,
And this
because
is
I
more
can still,
analogous.
yeah.
Yeah. And this is more analogous to a ligand binding a receptor because once the thing binds, it's not going to undergo any sort of reaction.
Okay. That makes ooh, excuse me. That makes
Oh, you're good.
sense.
Yeah. So yeah. So it's like it's a simpler system than KM.
Okay. Never mind. I
What were
answered
you,
my own question.
are you sure?
Yes.
Okay.
It would still remain the same across all inhibitors. Like, yeah, I mean, KD would change, but
Right.
it's still, yeah, the concept remains for all inhibitors.
Yeah, yeah. Yeah, and it makes sense that the dissociation constant, it's not an association constant. So it makes sense that it'd be 1 over affinity.
Mhm.
Also, actually here, okay, this makes it even better. This is really going to get to the bottom of this concept without even having looked it up because and this kind of shows you that you can just think through this stuff. So you've got like let's just say where I'm going to say ligand and receptor so that we don't have to think about an enzyme because that's kind of like a red herring here in this case because we're just talking about the ligand binding the receptor. So you have actually, and I'm just going to say L and R. All right, so you have L plus R, ligand and receptor, and that's going to be at some equilibrium with your like ligand receptor complex, right?
Mhm.
Because they're going to bind and they're going to fall apart sometimes, right? And
Mhm.
so you can write an equilibrium constant for this reaction. And your equilibrium constant is your products, right? Divided by your reactants. So concentration of independent L and independent R.
Right.
And your dissociation constant is literally just 1 over your KEQ of this reaction. Because the KEQ is telling you how much you favor the products here. And so dissociation constant is like how
Would be
much do
the
we favor
opposite.
not the products, right? Yeah.
Oh, okay. Yeah.
That's where that comes from, I think.
Cool.
So
Yeah.
just like reasoning through all of it. And I'm
Cool.
guessing C is the right answer. I haven't clicked the right that is the answer yet,
No,
but
I don't even
the one that's
C is in fact the right answer.
okay, yeah.
The distribution was pretty even. It was like 23-24% on each answer.
Oh, really? Nice. Yeah, I'm
Yeah.
just gonna go ahead and click this. That way I can see what all of them are. Uh 1 second, sorry. This is why I didn't do it yet. Okay, there we go. Yeah, okay, yeah. Yeah, so 34% got it right. That tells you that a lot of people guessed.
Yeah.
Like, very few people knew that because that's a hard question.
Honestly, it was a really short passage, which are my favorites. But it was
Yeah,
jam-packed with anytime it's jam-packed with a lot of acronyms
yeah.
of something, I get lost.
Yeah, yeah,
It's
that was my issue when I was taking the MCAT also. It's just like, yeah,
but that's like
just
literally all of biochem. It's so many
yeah,
acronyms.
I
So I need to
know.
get past that somehow.
Yeah, so much of it is just gonna be it's more I think that's more a confidence thing than anything else.
Mm,
That if you just, like, go in and be like, "Yes, screw you guys, I know everything," then that's gonna be less of an issue.
I think so.
Yeah.
I think my other thing is like, I mean, on paper test at least, I usually like highlight or circle certain things
Mhm.
as I'm reading
Yeah.
because otherwise I like lose my train of thought or whatever
Yeah,
it is.
same.
And I like can't really do that here. I like
Yeah.
really wish I could just draw on it somehow, but
I know, that's my issue with that kinda stuff as well, and in my med school didn't help any because most med schools will do, like, virtual tests now,
yeah,
also,
I
like, you don't
figured.
take any paper tests, but mine did everything with Scantrons and, like, paper tests and stuff, except 3rd year, then 3rd year, you have to take everything on the computer. So, you might as well just get used to this because this is how it's gonna be from here on out.
Never in my
You
life did
may
I say I would miss Scantrons, but I miss
yeah.
Scantrons.
Yeah, you may never see another Scantron
No,
in your life. I mean, we
that's
used them, but that was also a while ago. Like, I started med school in 2013, which is a really long time ago now that I think about it.
that's really disappointing as someone who does everything on paper, but that's
Yeah, no,
okay.
I look like somebody that's very, like, computer oriented, and I guess I've become that, but not by will, just by, like, knowing that I need to, because I don't
Fair
like
enough.
I print everything out still, which is a huge
Yeah,
waste of paper, but I like to have hard copies of things in front of me.
they're gonna make the paper anyways.
Yeah, I like it. Yeah, they're gonna make the paper. Might as well just get the GSM 250 and then like that way everything stands up on its own.
There you
Do you
go.
know what I'm yeah, if you get do you know what GSM means? Well, sorry,
GSM
this is a huge
250.
digression. Yeah.
Huge difference. Isn't that a monitor of some sort?
No, so GSM is a paper rating, and it's grams per square meter of the paper. And so the more
Oh.
GSM you have, the like thicker the paper and like nicer the paper is. So if you buy
Whoa.
GSM 250 paper, it's really fun to have, but it's expensive. So it's kind of a waste of money. I wouldn't buy it just for like printing stuff out. I only buy it for like flyers and stuff.
Interesting. Okay.
Yeah.
Now I'm gonna
Anyway,
explore paper.
yeah, huge digression again. But all right.
Huge
So
digression to, I don't know, other tangents. I think that's it for me,
oh, that's all you have.
on the questions. Well, I mean, I got a lot wrong. I would say roughly 50% of my questions I got wrong.
Yeah,
But
that happens though.
yeah, the explanations were pretty self-explanatory.
Right.
Um,
Yeah. Yeah. That's why
and
I
there
like
were
it well. They're
repetitive themes. Like the enzyme stuff, I like consistently kept getting the type of enzymes wrong, which was really stupid on my part because I literally read those explanations and could have answered them correctly the next time, but I didn't. Um, so yeah, there I now have started to recognize some themes and like gaps and such. But
Yeah, yeah, if I were you, I would just, like, make and every now and then whenever you're making like, writing some of the stuff down, I would keep some of it, and, like, for instance, one paper that I would write down and keep is, something that has the definition of, like, the zymogen and the proenzyme and whatever, like, else, the, like, apo enzyme, holo enzyme, coenzyme, cofactor, all of
right, right,
that stuff,
right.
and just, like, define each of those for yourself and then you can have that and refer back to it because I don't know about you, but that for me is one of those things that it's like I just always forget. Like
Yeah,
I always look that up whenever somebody asks me, just because I have notoriously forgotten that constantly, like over years of knowing it. So yeah, that's why I googled that stuff while we were talking because I just wanted to make sure I don't want to tell you the wrong
that's
thing.
fair.
Yeah, and that kind of stuff is stuff that you don't like. They're gonna try not to make you miss a question because of that. Because they wanna see you think through things, like, they want to test your ability to do, like, this, like, to come up with this
Mhm,
without without an essay, which is kind of hard to do, but that's what they're
mhm.
trying to do more so than, like, see if you can memorize what an apoenzyme is.
Fair enough.
Yeah.
Well, now I know.
Yeah, nice.
Yeah.
Okay, cool. But yeah, so I think so if that's all the questions you have, then we can be done early today. Which I'm totally fine with. If you think of other questions, I know you won't, but send them to me
Yeah.
and I can send you stuff back. I will right now send just write up really quickly the step by step of that one mechanism just because I want you to have that because I'd kind of forgotten a lot of it the other day and I just want
Yeah.
to make sure that you have it. So I'll send it
Briefly
to you.
before we go,
Uh-huh.
what would you so I drilled biochem and obviously I'm not doing well enough or like not how I want to be doing on this.
Mhm.
Do you recommend continuing to drill that or start mixing it with other subjects, even though I'm not like 100%
How many
well,
questions do you have left on it? On a total in
on
biochem?
just
Because that's
biochem?
yeah, I just don't want you to run out of biochem
Oh,
questions with,
fair, fair,
like,
fair.
a month to go or something.
Yeah, yeah. Let me take a look. I have, oh yeah, I have 147.
Remaining?
Yes, of
Okay, I would start
biochem.
mixing it in with other
Biochem.
stuff then. You could also go back
Oh.
through and redo the questions that you've already done.
Oh, I plan on
Um,
it. Yeah, I
yeah,
definitely do plan on it. Just to make sure that I can like I can.
yeah.
um,
Plus, there's gonna be stuff like this. It's not like a thing where if you just like do all the biochem questions, you're gonna get all biochem right. It's there's gonna be stuff in the other sections that are gonna kind of like jog your memory on some biochem stuff and give you more tools for answering the biochem questions, right? So I tend to be a proponent of kind of mixing it together. That way, everything it's like a rising tide that lifts all boats, sort of. Just like your
mhm.
total MCAT knowledge, but and that way you don't run out of biochem questions. That said, if you want we can just I can also give you access to my Kaplan question bank if you don't tell Kaplan. Because I have like 6,000 unused questions on there. There's a shitload of questions, and I'm sure you can separate them out into biochem questions. I haven't used any of them. I've just been using the only reason I bought that is so I could have the Kaplan books. I think you like yeah, I was talking to you the day that that happened. Um, so I have
Wait, does
all of
that
that
mean
stuff.
I have a
Oh
Kaplan
yeah.
books?
Yeah, so
I
you can
have
use their biochem questions also.
what?
Let me look.
And I
I'm
haven't
pulling
known
mine up
about
right now.
this?
Uh-oh. Welcome back.
I opened Kaplan and it immediately shut you down.
Oh, nice. Yeah, so you should have access to with Kaplan. Did you get access to like the online portal when you bought the books?
Yeah, I have this
Yeah.
portal.
Yeah. So there's a lot of questions here. See the thing that says QBank right here? If you click that, there are this many biochem questions. Yeah, so you could also use those.
I have literally had this for so long, and I never
Yeah.
noticed that. I've done like their like little learning path. I started. I mean, I haven't finished all of it, but they have like their
Mhm.
practice biochem. But I realized it was the
Yeah.
same as the ones in the book. Um, that's crazy.
I'm just
Okay, I should actually do these. What the heck? Wait, I don't have the Q bank. This is lies.
oh, okay. Well,
You have the
I can
Q
give you my
bank.
log if you want it. I don't care. I was just seeing how if they give you because what I remember of Kaplan when I worked for Kaplan is that
Mhm.
they don't have good explanations to their answers, which is why I tell people to use the UWorld stuff instead.
Oh, that's
Um,
so fair. I still have for now, I'm gonna say I'll stick to the student one because I still have, like, these practice ones that they have up on their, like, learning path or whatever.
okay.
I still have some of those, but, um, I may ask you for more. Indeed.
Yeah, so I guess it looks like this is a little bit more than they used to give. Their explanations used to be like this long
Maybe they
for
just got a bunch
each of
of
the questions.
really bad Yelp reviews.
Probably, I wouldn't be surprised.
Yeah,
Yeah, that looks a little bit better than what they used to do. So yeah,
cool.
if you want, just let me know and you can use all of my Kaplan stuff. I don't care, just don't tell them
Okay.
because they'll sue me and that would suck.
We'll personally call them up.
Yeah, they'll sue me for both of my dollars.
Okay, cool. Do I mix in the meantime with like specific subjects or just go for like click
Mm.
all of them and see what happens?
I think I kind of I'm biased toward the clicking all of them and see what happens because that's more like what the real test is gonna do to you.
Okay,
That'll
cool.
also like move you through the content at somewhat of a similar pace. That way you don't run out of any one thing. But if you're feeling weak in one area, then you can just do that also. Also, don't forget that the Kaplan books themselves have a bunch of questions in them at the ends of chapters that you probably haven't done yet.
Mhm, mhm.
They're not like MCAT style questions, but if you're just wanting to be, to assess yourself on knowledge of some of the stuff, then they work for that. So
Okay,
that works as
yeah,
well.
I will
Yeah.
definitely do that.
And you can also use more UWorld biochem questions if you want. Now that I'm thinking about the fact that we have these Kaplan questions, I'm not as worried about you running out. So it's totally up to you,
Cool.
really. Yeah. Which
Yeah.
I'm not meaning to punt on every question. I just don't like telling people telling people specifically what to do if, in my opinion, there's not like a best option. So it's kind of whatever is just like I said the other day, the path of least resistance. However you're gonna get yourself to do it, just do that.
Yeah. No,
Yeah.
I agree. I think I'm gonna try mixing, and then if I notice something that's like a gaping hole in my knowledge, I'll focus on that. But otherwise,
Okay.
yeah, I think that's a good idea.
Yeah, and if you notice something that's a gaping hole, also you can just let me know. We can schedule an impromptu meeting as well. Like I really don't have that many customers right now, so I'm very free most of the time. I'm just working on like making videos like the one I sent you earlier. So yeah,
Cool.
yeah, those
All right.
whiteboard videos suck. Um, but anyway, so yeah, just let me know if you have any questions, if you want to schedule a different meeting. Otherwise, I'll just put in. Is it cool with you if I just put in for next Tuesday, same time? Because
Yeah.
I noticed that we just didn't have any more meetings in the schedule at all. That's why I sent you that
Oh, yeah.
story.
Thanks for catching that. I did
Oh
not
yeah, no
notice that at
problem.
all. All right.
I mean, I was planning on meeting with you today at 4, and then I got on and noticed that there wasn't one. I was
Yeah.
like, oh no.
It's in
Uh,
my head. Like I know my
yeah,
schedule
yeah,
there.
yeah. It's in my head, in my calendar in my head, but that's
Yeah,
not
yeah.
that reliable sometimes. So better just to
Very
have
true.
it on here. So okay, cool. Um, so you're good for now? No other
Mhm.
questions or anything?
Yeah, still sticking to 30 a day.
All right, cool. Um, I actually don't have a good question to ask you on skis because I've just started looking. But uh yeah,
If you
so
do, let me know.
okay, yeah, I'm trying to
Um,
just I really don't know enough to even ask questions. That's kind of my issue right now. I'm just trying to see, because I'll just go to the rental place and be like, I want the most aggressive skis you have, and they'll give me good skis. And so
nice.
I have to figure out what that actually means in terms of
Nice.
like, yeah, all of that. So anyway, cool. I will talk to you on Tuesday, if not sooner. Then.
Sounds good. All right.
All right,
Bye.
cool. Talk to you then. See ya.